Hello students, today I am going to teach about culture preservation. So the methods of the preservation can be that agar slant culture is being covered with the oil and then it undergoes into a saline suspension and then it is being preserved at a very low temperature, and then after that it was being drying in the vacuum, and then lyophilization or freeze drying is being done. Then we come to a technique known as agar slant culture. Agar slant culture is being prepared in vitro, and after inoculation, slants are incubated for a period of 24 hours. Then it was being stored in the refrigerator, and the culture required periodic transfer after 6 months. You can see that we have a stock culture, and this is the culture tube, and where the bacterial growth on the surface media is there, and then you can see that uninoculated tube of the media, where the media is first prepared and then the bacterial growth is being done. Agar slant culture covered with oil. The agar slants are incubated after inoculation until profuse growth appears. These are then covered with sterile mineral oil to a depth of 1 centimeter above the tip of the slanted surface. Transfers are made by removing a loop full of growth, touching the loop to the glass surface to drain off excess oil in the medium, and then preserving the initial block culture. So we can see here the nutrient agar medium and some samples. What are saline suspensions? These are high concentration of sodium chloride, which is used as inhibitor of bacterial growth. Bacteria are suspended in 1% salt solution in screw cap tubes to prevent evaporation, and these tubes are stored at room temperature. Now, preservation at very low temperature. Like the organisms are suspended in nutrient broth containing 50% glycerol or skimmed milk containing 7.5% of glucose. These suspensions are frozen and stored at minus 15 to minus 30 degrees Celsius, and ready availability of liquid nitrogen at minus 196 degrees Celsius has provided another means of preservation of stock cultures. Now, preservation by drying in vacuum. So, organisms are dried over CaCl2 in a vacuum, then stored in refrigerator. These organisms survive longer than when air is dried. So, what is lyophilization? Lyophilization is the microbial suspension is placed in small vials. Then a thin film is frozen over the inside surface of the vial by rotating it in a mixture of dry ice or alcohol at the temperature of 78 degrees Celsius. Then these vials are connected in high vacuum lines, and then it was being dried or frozen. The ampoules are then sealed off in a vacuum with a small flame, so that it cannot get contaminated. These cultures can be stored for several years at 4 degrees Celsius temperature. So, to revive microbial culture, it is merely necessary to break up the vial aseptically, to which a suitable sterile medium is added. The process permits the maintenance of a large number of cultures without variation in the characteristics of the culture. which generally reduces the danger of contamination. So these are some of the steps you can see here. And this is the diagram of the vacuum chamber from through which this process is goes. So we can see that this is a vacuum chamber. Upper to that is a vacuum gauge where pressure is being controlled, and this is the vacuum release valve, and these are the condensers where the ice is being placed, and cooling coils are there to cool, and regulator is there, and there is a vacuum pump and vacuum gauge which provides a vacuum medium to this device. Stock culture collection centers. So there are some of the collection centers, namely American Type Culture Collection USA, Indian Collection of Industrial Microorganisms Pune, Institute of Pasteur Paris, Microbial Type Culture Collection Japan, so Institute of Microbial Technology Chandigarh, National Collections of Type Culture London. So these are some types of stock culture collections. Thank you very much.